And now we come to a title which is really challenging. Shmuel Muli ben Sasson, you are in the Department of Development Biology Cancer Research at the Hebrew University and Medical School of Hadassah, Jerusalem. And when we talked about you, we said it must be like in life, when you want to bring something in order, you have to see the failures of the past. And when I asked you then to speak about learning out of the failures of the past, a new strategy for cancer, you consented that you would talk to this today in Basel and put some things that are straight upside down, I have heard and read.
I have to have an appetizer for dinner, so, okay. So thank you very much. So cancer research is undergoing groundbreaking transformation in recent years. No more individual labs, each working on a PET oncogene or tumor-suppressor gene in a subset of tumor clones, but rather a concerted effort once and for all to crack down on the profile of human cancer genome in a comprehensive way and in an unbiased way. And this culminated in this project known as the Cancer Genome Atlas. It's really an industrialization of the effort, industrialization in the best possible way in terms of standards, in terms of efficiency, and in terms of quality. And the project started 10 years ago. It was concluded this year. During this 10-year period, around 11,000 different samples of human tumors representing all major human cancers were collected, and they were analyzed layer by layer for mutation, copy number, gene expression, DNA methylation, microRNA, protein phosphorylation, clinical data, in a hope to boil it down to some simple thematic way that will enable us to design rationally a treatment for cancer. So after this project, no one can say that we are lacking data. I mean, there are details that are still missing, but we have the bulk of the data to evaluate it. The working hypothesis behind this tumor cancer, the Cancer Genome Atlas, was that cancer play chess, namely the distinct pieces, the genes, and if we will just recognize them, we will understand what is their function, and then we can design a counterattack against cancer. Unfortunately, it turned out that cancer is playing sheshbesh. I don't know if Basel you are playing sheshbesh, but it's very common in the East. In other words, cancer is pulling diabolic dice from its sleeve, right and left, and after such a strong statement, I have to justify it. So what have we learned from this really mammoth project? So there is a dramatic heterogeneity. In other words, intra and intertumors. If you take just a tumor, in this case pancreatic cancer, and you take pieces from different corners of it, you'll see that you have different clones. If you are looking at two different liver metastases, they're completely different, and of course they evolved further into a more complex genetically, and obviously the similarity between the genomic changes between cancer, this particular cancer between two different persons is quite remote. There are multiple mutation mechanisms way beyond what we used to know or think about in terms of point mutations. They are operating at high rate. We have a phenomenon called kataegis, that where you have a cluster of mutations. The original estimate of Vogelstein, the conservative one, that there are around 125 driver mutations, which means mutations that matters for tumor involvement across all cancers, and the more current refined analysis talking about around 500. And okay. Among those driver genes, the majority are not mutated oncogenes, which are druggable, but they are tumor suppressor genes, which are usually not druggable. Vogelstein tried still to make sense of all this collection of driver mutations, and if you look unbiasedly at what he suggested, it's everything. All major pathways can be involved in neoplastic transformation. So we heard a lot about personalized medicine, etc., but we have to realize that even if we accept that cancer is a signaling disease pathway, which I totally disagree with, but let us, for the sake of argument, let us assume that we have only oncogenes, and so you have this, and they are mainly intracellular. I mean, there are some cell receptors, but you have, you heard, we'll get back to it, you heard about BRAF, and you heard about MEK, etc., just by Professor Gerber, and so you see these golden rings, and these golden rings are supposed to be the specific inhibitors. Please be aware that these golden rings were discovered by high-throughput screening of combinatorial libraries, and it so happened that all protein kinase inhibitors are falling into the ATP pocket. And the ATP pocket structure, three-dimensional structure, is very similar between different protein kinases. And there are 600 different protein kinases in our body. ATP is ATP, and its pocket is optimized to accommodate it. So therefore, there is a built-in toxicity in this family of so-called biologic inhibitors. And we know, for example, that many of them inhibit around 10 and even 20%, those that are in the clinic, of all protein kinases. So we heard a lot about personalized medicine, and I would like you to distinguish between two distinct types of personalized medicine. The first is predefined target-based personalized medicine. In other words, we know that the mutation in BRAF in melanoma, but not in colon cancer, ALK, etc. I mean, we know that we have useful therapy there, useful drug, and therefore it makes a lot of sense. I mean, it is a must to do this screening for BRAF in melanoma, etc. You know, the way that in breast cancer we are staining for HER2. But there's another kind of personalized medicine, and this is what I'm calling fishing expedition-based personalized medicine. When you take a whole exome sequencing, it costs a fortune. It came really out of desperation of patients and the pressure that they put on physicians. Does it lead anywhere? So there is a subtext to this fishing expedition personalized medicine, that cancer is not a single disease. It is not even a collection of 100 or so different diseases by a tissue of origin. It is the outcome of random, endless combinations of driver mutations. Fine, c'est la vie. This is the end of evidence-based medicine. If you would like to do treatment by this fishing expedition medicine, I mean, namely, you have here, I mean, many companies would love, and actually there are many companies who are trying to build on this fragile population of patients and the relatives. So you send, and it's error of DNA, so you send the tumor DNA, enter it into a black box. Behind this black box, you can put many different names. And then at the end, you'll get pills and bills, but you will not be able to assess whether it is useful or not. And it is, I know that you might say, okay, these are facts of life, but the temptation for all sorts okay, I will not speak about those who will attempt to take advantage of it. So what we have at the patient bedside at the moment, with current pipeline of targeted small molecules inhibitor, meaningful therapy is achieved either for a short period of time or for a few patients. I mean, again, these are you can really count it on one hand. I mean, crizotinib or Glivec, etc. I mean, such outstanding successes, but it doesn't hold to the majority, and there is it is associated with collateral damage. If you add a BRAF inhibitor, I mean, if you complement it with MEK inhibitor, etc., you have to take into account what it's. And again, I'm talking about this type of molecules. I'm not talking about monoclonal antibodies. I'm not talking about genuine biologics like tamoxifen, etc. These are true biologics. I'm talking about the rest, and unfortunately, the majority of target driver genes are intracellular. So okay, so you saved me any further explanation about this this amazing picture. It's a miliary melanoma, as you heard. And you see the spread of these nodules, okay, all around. And after 15 weeks on vemurafenib, it's a miracle. It's unbelievable. I mean, it's outstanding. After additional 8 weeks, it comes back. And if you look carefully, it comes back exactly where the original lesions were. Okay, so you have like this here, you have this here, etc., etc. Okay. So, how such outstanding victory was turned into a tragic defeat? I mean, again, the patient might say, 2 months or 3 or 4 are a lot, and nobody can judge. The question is why, and the answer is because the occurrence of multiple mutations and aberration in cancer is obvious. So to conclude this part of my talk, my personal view is that there is no clean solution to a dirty disease, dirty from a genomic point of view. You might phrase it differently, cancer is a moving target. And the important question is really, can we do better? I would like to talk about two indirect strategies. I'm bringing up angiogenesis or anti-angiogenic therapy because I think it's a challenge for nanomedicine. I mean, you have blood vessels, and blood vessels, especially in tumors, they have special features, and therefore it was pursuing what we did in order to tackle with angiogenesis is, instead of looking for a single of a monotherapy, the way that the pharma industry is used, like Avastin, okay, anti-VEGF or the like, we said angiogenesis is a multifaceted process. Let us take this complexity to our advantage and build a drug combination that will synergize with each other and therefore will be much more useful than Avastin. And indeed, we were successful in it. I mean, this is just a case that was so dramatic in our initial trial of all comers of a sarcoma, a patient that was refractory to conventional chemotherapy that underwent a complete regression, and we are now concluding a phase 2 trial in pancreatic cancer. But I would like to devote the rest of my time to another strategy, and that is ignore oncogenes and tumor suppressor gene and attack the epigenome. And we tend to overlook the meaning of the epigenetic chaos to this phenotype of cancer. So aneuploidy and chromotripsis, I'll explain in a second, are very early events in pneumotumors. And even more so, if you take and you do mutations deliberately in genes which are responsible for the integrity of the epigenome, you will get instantaneous tumors. Usually when you're inducing tumor, it takes several months. I mean, it's a gradual process, etc. But if you're doing, and here you have several examples, when you are introducing a mutation in a gene which is in a protein which is in charge of epigenetics, you get instantaneous tumors, and we have, I mean, this really a fascinating example that it's a rare tumor, a rhabdoid tumor that just a mutation in this gene is causing transformation, instantaneous transformation in humans and in mice. So this is how real-life tumors look like. So SIN stands for chromosomal instability, and MIN stands for microsatellite instability, and shakshuka in Hebrew is scrambled egg. And real tumors do not look like point mutations in driver genes. Real-life tumors have a whole variety of copy numbers away from the diploid, the normal diploid, with a lot of translocations. This is microsatellite instability. We'll not be able to get into it. Chromotripsis is a shattering of a chromosome. If this was the order of genes on this chromosome shattering lead to this. And we know that there's the three-dimensional structure, the three-dimensional organization of the chromatin inside the cell means a lot for the ability to control gene expression. And indeed, when pathologists are looking at tumors, they are not looking, I mean, maybe now they will start to, but when they are doing diagnosis, they are looking at nuclear pleomorphism. And just by this nuclear pleomorphism, they do the diagnosis, and there is, think how much is standing behind this change in the three-dimensional organization of the nucleus. So, so I would like to suggest that from an epigenetic point of view, the majority of cancers are single entity, sharing a common denominator called epigenetic chaos. Now, epigenetics comes in all flavors. I mean, it's not just histone acetylation or deacetylation, lysine methylation, etc. It also has to do with DNA folding, spatial arrangement of the chromatin, EU versus heterochromatin, non-coding RNA, etc. So although for a person, if you look, if you give a closer look at a histological picture of a tumor, you recognize there are a lot of cell death ongoing. So the tumor is not that robust as the way that we tend to think. So although cell multiplication is overpowered cell death, tumors are on the verge of collapse in many sense, especially when it comes to their epigenetics checks and balances. And actually, in my view, what oncologists are doing for many years unknowingly treating cancer is by generating extra chaos. So you have the initial chaos, no matter how you look at it, each chemotherapy treatment is inducing extra epigenetic chaos into a genome which is already chaotic, and it can get to a point where the chaos is such that the cells are undergoing apoptosis. So even cancer cells can undergo apoptosis, and we know now that many of the chemotherapies are leading to genuine apoptosis. So here is my suggestion for anti-cancer selectivity, taking advantage of this insight of the epigenetic chaos of cancer cells. Normal cells have a solid system of epigenetic checks and balances, and cancer cells, in order to become cancerous, got rid of them. And therefore, if we just induced an extra epigenetic chaos, I know that at the moment it's out to your hands waving, but we are thinking in a very concrete terms how to do it in a controlled way. So the idea is to do it in a controlled way, and while tumor cells would be vulnerable and might collapse, normal cells have enough checks and balances to stand this shakeup. And luckily, the toolbox of epigenetics is very simple from a chemical point of view. So we are talking about small molecule modification. We are talking about methylation, just adding a methyl group to CpG. We are talking about acetylation. We are talking about methylation of lysine. So we can think about small molecules that can enter the cells and help introducing this increment of extra chaos in order to kill cancer cells. So in conclusion, contrary to the cancer genome, to the genomic perspective, the majority of cancer can be viewed as a single entity, unified by epigenetic chaos, and accordingly we should develop new tools for a new era, and that's what we are trying to do. So thank you very much.
Thank you very much. Thank you very much. And first questions? Yes, Simon.
Muli, thank you very much for this enlightening but controversial lecture. Sure. I would like to ask you, for years we have been told about the plasticity of the DNA of cancer cells, the karyotypic instability, all these leading to the fact that very quickly cancer cells develop resistance even to the best designed targeted treatments we can give to our patients. So what is, now you are putting all this on the epigenetic control of the cell, and I was wondering, so what is the cause for the fact that we have, after all, we know that there is gene predisposition for cancer, there are mutations that go and from mother to or from parents to children, and there is a whole thing built about like a threshold number of mutations that will indeed cause cancer. And this is a very ordered way to look at this building. And what you are bringing up, although I think it is very attractive, because we may find ways, if we address it in this direction, to treat many types of patients and not to believe that we can develop an individualized treatment for each patient, which I agree with you is really absolutely, it's not going to lead to a way to be effective in public health. But back to the basics, how do you reason this epigenetic chaos from the origins of cancer as we know as gene mutations?
Okay, it's an excellent question, Alberto, and the answer is simple. It's simply our awareness in recent years that there is a very strong component of epigenetic control of gene expression to the three-dimensional organization of the chromatin in the nucleus. Because for years we used to think that every gene is an island, isolated. So you have the gene with its control elements, and we know now that changes, for example, changes in transcription factors can change the whole three-dimensional organization of the chromatin, and different regions start to interact with other regions. So technologies like the high C, etc., help us to realize that there is a component of a change of epigenetic chaos to these chromosomal changes, etc. So they are not just you have an extra copy of this gene or other gene, but you have a whole chaos in terms of the control of gene expression. So this is, and therefore it would be just, you can take and add to this, you can now add well-controlled modulation of the epigenome, in other words, further impairing its activity, and you have, for example, in PML, okay? In PML, you can drive PML either to differentiation, or you can drive PML into apoptosis by oxid,
One of the HDAC.
no, not the JAK inhibitor, the Chinese, the old Chinese salt oxide, not aluminum. Okay. Arsenic trioxide. All right, arsenic trioxide. So arsenic trioxide that can do this miracle of when you look at the effect of arsenic trioxide on cells, you can see that it changed many components, including methylation, cytosine methylation, etc., in a way, and now you can see how it can help lead to differentiation or apoptosis, which is exactly what you want.
So, Simo.
Yes, I was wondering in the study, this huge study that you show with different omics applied to hundreds of different tumor specimens, how do you distinguish what's a bystander alteration from what is not bystander? And I mean, we have several examples, like in microsatellite instability tumors, those tumors in which there are mismatch repair deficiency. You'll find hundreds of mutations, and only very few of them probably are driver mutations. So most of them are just bystander. So I'm wondering, in those cases, how do you manage to do that?
So, right, I will not be able to go into details, but there were rules or methods that were developed as part of this project to being able to distinguish between driver mutation and what is called passenger mutation, or bystanders mutation. And there are many of them as part of this chaos. So Vogelstein has this rule of 20-20. I mean that if it's in a certain point of a gene, in at least 20%, etc. And there are all sorts of rules how to, and he was more conservative, and then more sophisticated statistical methods were developed, and the estimate went up from 125 in his hands to around 500. But there's a whole literature about how to distinguish between between real driver and passenger mutation, because there are much more passenger mutations than driver mutations.
Simo, Jacinto.
Hi, my question is about, on a cellular level. There are many claims that autophagy is a kind of mechanism that is encouraged to stabilize cells when some chaotic phenomena happen on the cell level. Can you comment what could be the autophagy role in this anti-chaotic kind of medicine, if you wish?
Autophagy is considered, actually, as far as I know, as a mechanism to protect cells from collapsing. In other words, to reserve the ability of the cells to survive in case of deprivation. You chew some of the extra organelles. I mean, you move down to a low level of activity and avoid it. So it's.
Jacinto.
What you suggest makes a lot of sense, but why to neglect the third way, concentrate research on early diagnostics?
Vogelstein published some enlightening articles on this. Of course, early diagnostics is the best. Not enabling people to smoke is a very important thing, and early diagnosis is the way to go. And this is, part of his lessons, similar to part of his lessons of this. Part of Vogelstein lessons from this picture of the realization of the inability when a tumor is in an advanced state is let's invest all of our efforts in early diagnosis. So you're absolutely right. So this is another lesson of this.
When I look into the hall, I feel that everyone is now so hungry that we maybe do in a bilateral the next quarter of an hour, we skip it and would go for dinner. We will at 7:30 meet in front of the Hotel Le Plaza. You are all cordially invited to be there then. We have for 7:45 reserved a tram which brings us non-stop to Rien. I also wanted to say that although tonight the train will go back on 23 something, we would really start tomorrow, nevertheless at 8:15 as it's in the schedule, and we count on your accuracy to be here then. Thanks a lot.
There's a young lady who wished to ask.
The young lady has to ask another question.
I'm sorry, it will be a quick one, I promise. Thank you for your talk. I wanted to ask you about the selectivity you said for normal and cancer cells. You mentioned that it's based on like the epigenetic control. But I would like to ask you if like any other parameters could be involved in this selectivity. Could it be like a structure that a cancer cell has and is not found in normal cells, like glycocalyx? We know that is a structure that is much thicker in cancer cells. So could this be responsible for the selectivity you mentioned?
But you are taking it to a different, to something that is dated 80 years or more, and this is to define something that exclusively distinguish a structure, an enzyme, whatever that's distinguished exclusively between normal and cancer cells, and there is none. We can argue about glycocalyx as well, etc. So and mucins, etc. But it's and therefore I was suggesting this strategy from within.
So many thanks.